We will move on to the next class. Up till now, we have seen the synchronalkaloids and we have seen the 4-aminoquinolines. Next class is the 8-aminoquinolines, again related to the quinoline ring. You have the quinoline ring, that is the benzopyridine ring constant. And as usual, we are going to start numbering from the heteroatom, that is nitrogen, and complete the ring. So this is first position, fourth, fifth, sixth, seventh, and eighth. So at eighth position, you have the amino group. That is why this class is called as 8-aminoquinolines. Now here is array of structures of the drugs, antimalarial drugs, which belong to the 8-aminoquinolines class. The first member of this class was Parmaquin, but because of the different, what you can say, issues that were associated with Parmaquin, now if you see in the market or in the clinic, it is only the Primaquin which is used as a antimalarial agent. The other drugs obviously are there, Pentaquin, Tefnoquin, Bulaquin, Quinoside. These are the newer analogs. But traditionally it was Parmaquin, Primaquin, Pentaquin, and Isopentaquin. The Tefnoquin, Bulaquin, and Quinoside are the newer analogs. Now in our syllabus, we are going to focus only on the Parmaquin and Primaquin. So as I told you, the first compound introduced was Parmaquin. You can very well see it is an 8-aminoquinoline, and in addition to this substituent at 8th position, substituent, the alkyl substituent on the amino group at 8th position, you also have a methoxy group. Where have we seen this methoxy group, you remember? Which drugs the methoxy group was present?
Quinine and synconidine, quinidine, right?
Quinine and quinidine, all right, because synconidine was a desmethoxy analog, as well as synconine was the desmethoxy analog. So definitely quinine and quinidine were having this methoxy group. Okay, so the same is again retained or brought forward or brought into the structure of the 8-aminoquinoline. You can very well see, or you must have realized, that there is again just a single chiral center in 8-aminoquinolines as well, be it Parmaquin or the structure of Primaquin. What is the main difference in Primaquin and Parmaquin? Can anyone tell? Just have a look and compare the structure. What is the difference between Primaquin and Parmaquin?
The amine has substitution of ethyl group.
Yes, very good. So basically in Primaquin, you have a primary amino group, right? While in case of Parmaquin, it is a tertiary amino group which is present, because both the hydrogens of the amino group, they have been replaced by the ethyl group. So two ethyl groups have been brought into picture in case of the Parmaquin. So this was the first drug actually, Parmaquin. So if the question is the first 8-aminoquinoline, the answer should be Parmaquin. And the only drug which is there in clinic now is the Primaquin. Amongst the older analogs, it is only the Primaquin. And how it differs from 4-aminoquinoline? Simply because the group is present, the substituent amino group which is substituted, it is present at 8th position rather than the 4th position in case of the 4-aminoquinolines. Is it clear to everyone?
Yes, ma'am.
Okay. Now we will definitely be talking about the details of the structures, but for now you just remember that all of these 8-aminoquinolines, be it Parmaquin, Primaquin, they are associated with a specific, what you can say, side effect, that is hemolytic anemia, especially in patients who are glucose 6-phosphate dehydrogenase deficient. Now, this G6PD enzyme, it is very, very important for the proper functioning of the RBCs or the erythrocytes. And in general also, the patients who have this G6PD deficiency, they don't have the enzyme, there are chances of lysis of the RBCs. Okay, the RBCs will simply lyse. But if you are giving Primaquine to such patients who are G6PD deficient, you are increasing the risk of hemolysis. Okay, and because there will be too much of hemolysis that would be occurring in presence of Primaquine, mainly it's metabolite. It is not just the Primaquine. It is not Primaquine, in fact. It is the reactive metabolite of Primaquine, which is formed, and that is responsible to bring about the hemolysis. That is responsible to even aggravate the hemolysis problem, which is already associated with the G6PD deficient patients. And this finally leads to what is called as hemolytic anemia. Okay, it can lead to hemolytic anemia. So remember, Primaquine on metabolism, it will lead to the formation of a reactive metabolite, and it interacts with the uh, ion of the hemoglobin and leads to formation of methemoglobin, which finally produces what is called as a oxidative stress in the cell, and it finally brings about the hemolysis. And too much of hemolysis, it is finally going to lead to the hemolytic anemia. So your contraindication or the case in which you cannot give the Primaquine is the G6PD deficient patients. Okay, you cannot give Primaquine in G6PD deficient patients. In addition to this, even the neonates or just born babies, they do not have the well developed metabolic system. They don't have all the enzymes which we have today, okay, after years. So even in babies, it is very, very risky to give Primaquine, especially as a treatment for malaria. Same is the case with even pregnant ladies, because then that can travel to the fetus. Okay, and again, fetus obviously is not going to have the G6PD, so that is going to create problems. So these are the three stages or cases where you cannot give Primaquine as a antimalarial agent. Is this clear to everyone?
Yes, ma'am.
Yes, ma'am.
Yes. Okay. Now, when we're talking about Primaquine and Parmaquine, along with that, two structures, extra structures have been added over here, just for understanding, pentaquine and isopentaquine as well. The difference for Primaquine and Parmaquine, it is obvious. In case of your Parmaquine, you have the two ethyl groups, and in case of Primaquine, you have the primary amino group. But when you talk about pentaquine, you have a isopropyl group. One group is isopropyl, the other is retained. So basically, you have a secondary amine rather than a primary or tertiary amine, right? सेम इज द केस विथ आइसोपेंटाक्विन. यहाँ पे से पेंटाक्विन की स्ट्रक्चर देर इज अ प्रॉब्लम व्हेन आई विल share the PPT once again. I'll make that change. There should be 5 carbons between the 2 nitrogens. Okay, that is why it is called as a pentaquine. 5 carbons are there, so that one CH2 is missing from here. So you just have to add one CH2. Even in isopentaquine, you have the 5 carbons. The only thing is it is a branched compound, or you can say branch chain which is present. So it's isopentaquine. Okay, you have a isopentyl group. That is why it is called as a isopentaquine in case of the drug. Now, all these compounds, they are chiral except for the pentaquine. Okay, except pentaquine, all the other drugs, they are chiral. And these are the chiral centers which are marked by the asterisk. It is very obvious because these carbons, they are attached to 4 different groups, and that is the definition of your chirality. So with exception of pentaquine, all the other 8 aminoquinolines, the remaining 3, at least on this slide, they have one asymmetric carbon. Okay, so because the isomers, they don't have a significant difference in the activity, or none of the isomer is actually responsible for what you can say toxicity or preventing the metabolism. There is not much significant difference. So that is why these drugs, they are marketed as racemic mixtures. So to talk. Yes. So we were at the classification of the antimalarial drugs, and as I told you, for antimalarial drugs, the main prototypical molecule or the scaffold is the quinoline ring or the benzopyridine ring. Okay, and uh, the first three classes as well as the quinoline methanol class. So these four classes are the ones which contain the quinoline ring scaffold as a part of the drug, and there will be certain modifications as well. Then among cinchona alkaloids, it's the quinine, quinidine, cinchonine, and cinchonidine. As I told you, we are just going to focus mainly on quinine because that is the one which is given in syllabus. And the others we will be just talking about because they are related as far as the structure is concerned or the stereochemistry is concerned. Then we will talk about 4-aminoquinolines, chloroquine, amodiaquine. I've told you the use of hydroxychloroquine. Though it has anti-malarial activity, but it was repurposed for the COVID treatment as well. However, now it is discontinued for the same. Then amongst the 8-aminoquinolines, we will be focusing on Primaquin and Parmaquin. Parmaquin was the first of the 8-aminoquinolines which was introduced, but then now the mainstay is the Primaquin which is available. Then we will be focusing on quinoline methanol, that is a quinaquine. It's a new class again, which contains the quinoline ring. And of course, other classes as well, they are important. For example, the biguanides, in which proguanil, or it is also called as chloroguanide, and cycloguanil. So proguanil is the prodrug and cycloguanil is its metabolite, okay, active metabolite. Then we will talk about the diaminopyrimidine, that is the pyrimethamine, which is a very well-known DHFR inhibitor. So it is going to interfere with the folic acid synthesis in the Plasmodium species. Among sulfonamides and sulfones, we will focus only on sulfadoxine as such. Then you don't have amino alcohols specified, any of the amino alcohol drug in your syllabus. But as a class, you should know that it is used as a antimalarial agent. Next, this is a newer addition. Nowadays, it's the artemisinin combination therapy, which is generally used for malaria. So in this, we will focus on artemisinin. That is the main compound which is isolated from Artemisinia annua. That is the plant, the leaves of Artemisinia annua. And then its modifications, the artesunate and the artemether. So these three compounds, naphthyridine, spironolidine, not there again, and naptoquinone. This is a polycyclic compound, you can say, that is atovaquone, is something which we will talk about. And then again, you also have certain antibiotics which are repurposed or used in malaria, which includes tetracycline, doxycycline, and clindamycin. So remember, for this, we are just going to look at the drugs that are in red font. And for synthesis, it's only the chloroquine and the Parmaquin which has been mentioned. Okay. Any doubts in this?
No, ma'am.
No, ma'am.
Okay. So coming to the sites where these drugs act, now that we know that these are the different classes of drugs that we are going to focus on, so we should also know that which site they are going to act on. Okay. So when we are talking about the sporozoites, you know the mosquito basically injects the sporozoites in the humans or releases the sporozoites in the humans, and then the sporozoites, they basically go and attack the liver cells, that is the hepatocytes, and there are various conversions that are there. Some of the sporozoites, they could go into the dormant stage. Dormant stage is the one during which there will be no multiplication or there will be no conversion from one form to another. That is called as the hypnozoid. So this is the dormant stage of the sporozoid, as well as the other conversions which we have seen, the schizones and the merozoites. So all those conversions, they occur in the exoerythrocytic stage, that is in the liver. Okay, so you can have two types of drugs acting on liver, two types of antimalarials. The ones which act on the dormant stages of the sporozoites, and the one which acts on the non-dormant, or the ones which are in active replicating form, or multiplication goes on, or the conversion goes on. So two types of drugs we can have. So when we're talking about the antimalarials which act on the dormant stages, it is mainly restricted to the 8-aminoquinolines. That is, nowadays it's the Primaquin, and this is a new drug, tafenoquine, which is, again, we don't have it in the syllabus, but yes, we will be talking about Primaquin definitely in details. And then for the converting, this thing, what you can say, forms of the sporozoites, we can have either 8-aminoquinoline, that is a Primaquin, as well as the combination of atovaquone and proguanine. So this can also be used over here. Then when we talk about the erythrocytic stage or blood stage of the merozoites, basically, there it is the artemisinin combination therapy, that is the ACT, which is used, as well as chloroquine, mefloquine, primaquine. So all of the quinolines which we have seen, chloroquine belongs to the 4-aminoquinoline class, mefloquine to the quinoline methanol, and then primaquine. Primaquine is again 8-aminoquinoline, okay? As well as doxycycline and clindamycin. These antibiotics are also used in the erythrocytic stages. And then finally, at the gametocyte stage, where you have the immature gametocytes, and then they mature into the male and female gametocytes. So during this stage, you can use the quinolines, different quinolines which we have seen up till now, as well as the artemisinin combination therapy. ACT stands for the artemisinin combination therapy. Okay. Is this much clear to everyone?
Yes, ma'am.
Any doubts in this slide?
Ma'am, can you explain dormant and non-dormant in this in short?
Yeah. Dormant and non-dormant are the forms of the cells. Like, some cells, they are in active growing form, or they can replicate, but some will not replicate. They will remain as such, latent, what you call it as a latent form. Okay, so latent and dormant is one and the same thing. They would not be in the replicating stage. They would be non-replicating, or they would not get converted to the other form. They would remain as such. Like for this hypnozoid, if you are talking about, they would remain for long time in the hypnozoid form. They would not be activated to replicate further. They will not get converted to other forms. Is it clear?
Yes, ma'am. Thank you.
Yeah. So in liver, you can either have the dormant stages or the non-dormant stages of this sporozoites. So that is how you can have the drugs. But common feature is 8-aminoquinolines. They can be used against both the forms, dormant as well as the non-dormant forms. Yes. So to start with the drugs, the first class we are going to start is the natural products. Now in natural products, again, we can have cinchona alkaloids, as well as we can have the artemisinin type of compounds, artemisinin and its derivatives. But then being quinoline class, we will start with the cinchona alkaloids, which is a mainstay for the treatment of malaria. So in this, as I told you, we will be more focusing up on the quinine, but at the same time, because the other drugs are related to each other and they have significance definitely, we will talk a little bit about the other forms as well, though nothing will be asked in the university paper or the sessional exam with regards to quinidine, cinchonine, and cinchonidine. Okay. So first we will talk about quinine, and then I will just give you a brief, what you can say, a small introduction about the other drugs of the cinchona class. Okay. So you can very well see there is a general structure which has been drawn over here, and the main scaffold or the heterocyclic ring which is present is the quinoline ring. Now quinoline is nothing but it's a bicyclic ring which is formed by fusion of benzene ring and pyridine ring. So these two rings are fused and there's formation of quinoline ring. So quinoline ring is the main scaffold or the prototypical molecule in all the cinchona alkaloids, as well as the 4-aminoquinolines, 8-aminoquinolines, and the quinoline methanol class. Okay. Now, in cinchona alkaloids, that is all of these, quinine, quinidine, cinchonine, and cinchonidine, you have a linker, one carbon linker. Okay. What it is linking to? It is linking to another bridgehead or ring, or you can see over here, there is a ring which is having 2 carbons on the bridge. Okay. So this ring is called as a quin-nucleidine ring.